It would have been good for her to know that.
I'll send her the.
No, it's okay. It's too late. It's too late. Good afternoon, everybody. This is Martin Gershon, and we're here again with another BetterHealth series. We're talking today about precision medicine and drug discovery. It's an incredible new field, which is really going to change and shape the future of biotech. And it's going to be supported by AI and drug discovery techniques. Today, we are really incredibly thrilled to be with an outstanding company, Protein Fluidics. And the CEO of that company, Evan Comwell, is here today alongside Matthew Burrow, who is an incredible PhD scientist out of the Tulane Cancer Center, and Mahesh Narayanan, who is the director at Newchip, an incredible accelerator where I'm lucky to be part of the Series A investment team. I want to thank everyone in advance for their participation here. I am just thrilled week in and week out to receive these incredible emails from people who are talking to me about how these webinars have impacted their perspective on healthcare. And I am using this as a chance to highlight several different things within healthcare. First, the importance really of how innovation, science, and technology impacts human beings. What is the real impact on you on a day-to-day basis? And what we're going to be talking about today is one of the most sensitive areas of healthcare, which is cancer. And we'll talk a little bit about, first, what the current situation is of cancer treatment, and second, what the potential vision is of the future and what companies like Protein Fluidics are doing to enhance that, and what incredible accelerators like Newchip are doing to attract amazing companies, amazing entrepreneurs, and most importantly, leaders who can see the future, build teams, and build great companies through great collaboration with biotech, pharma, medtech, and health tech companies. So first, I want to also structure a little bit about what to look forward to in the next several weeks. Each month, we've broken down the BetterHealth series into 4 parts. One is an early stage company focus, emerging companies. Second is a focus on unicorns, where these companies have gone in terms of their growing and scaling and commercializing. Third, a very important focus of diversity, equity, and inclusion with respect and admiration to incredible CEOs who fall into the DEI category. And then finally, a high-level discussion once a week about what is happening in industry, what is happening with financing, valuations, what are the major sectors that investors are looking at, and what you can expect as a patient or as someone who's involved in health and wellness going down the road several years from now. With that introduction, I would like to first introduce Mahesh, who I think will tell you a little bit about himself and the work that he is doing at Newchip. So Mahesh, please let us hear about what incredible things you are seeing at Newchip, and a little bit of the personal story about your entrepreneurial experience, because you're quite an entrepreneur yourself.
Thank you so much, Dr. Gershon and Endeavor, for having me here today. As he mentioned, my name is Mahesh Narayanan. I'm currently the managing partner at Innovation Ventures, which is a separate fund, but also here representing our accelerator called Newchip, where I'm the program director for Series A. My background is mostly in neuroscience and oncology, so what we're talking about today is quite relevant to my own personal background. And I have run my own startups for a little over 15 years in the healthcare space, taking them from pre-revenue all the way to over $50 million in licensing deals and successful exit there. And this has also given me a very unique perspective on what to look out for in early stage companies, especially in life sciences, how to aid them through the potential pitfalls, help them hopefully to get to be a successful growth stage company and have exits of their own. And part of that is my involvement with Newchip Accelerator. And just briefly about Newchip, we are a global accelerator based out of Austin, Texas. I initially joined back in 2019 as an investor, and then later on as part of the team themselves, when we still had only about 4, maybe 5 people in the actual team. But we've had some tremendous success over the past couple of years. Fast forward to today, we have a little over 60 employees, not just in Austin, but all over the world. We work with a little over 2,000 startups, have a little bit more than 1,000 active investors in our network that we work with very closely, and a little over 500 mentors that we also are able to help pair with some of our companies that need help at the right time. And so it's been an absolute pleasure having been in this accelerator, having helped so many companies grow from that early stage, pre-seed, seed, all the way up to Series A. With the help of what we have is the network of experienced mentors, industry experts such as yourself, Martin, having been a part of the Series A team to help these companies and promote, along with Endeavor, the leadership, the entrepreneurship, what we are looking to really grow in the next couple of years. And just looking at the current macroeconomic climate, it's definitely a hard time to raise capital, but it's a massive opportunity for entrepreneurs. Downturns, recessions, they're often the golden eras, what we call the vintage years, the golden vintage years of companies that can grow and thrive and become the next, billion-dollar revolutionary companies that we're often seeing and hearing about every day. And so we are really looking to be able to do that for many of the companies coming through Newchip. And we've identified companies that have a very large growing market, have talented, experienced management team like Evan himself here, who's been through our Newchip program, companies that have really achieved next level of traction in the market, in acquiring customers, understanding the product that they really need to build for these customers, and these are the well-suited companies that we often like to work with and looking to grow with this partnership that we have here today. So I'm excited to be here, excited to learn from Evan the growth that he's seen since he and I worked together in the past, and also talk a little bit more about this very interesting space of synthetic biology, as well as how we can use this as a platform to build the next level of treatments, diagnostics, cures, and hopefully, we'll be talking about how we can do that for cancer today. So excited to be here. Thank you so much for having me, and looking forward to being part of the conversation.
Fantastic. Now, Evan, perhaps you can tell us a little bit about yourself and about the company, big picture, in terms of what you're looking to accomplish and where you see things going in your space in cancer treatment, in helping patients. I think you're muted. There you go.
Okay. Well, thanks for having me join, Martin. I started the company several years ago, really focused on providing new tools for scientists. My passion is really solving complex diseases and helping researchers like Matt, progress and do that. And the area of oncology is of particular importance to me, just personal experience in that area. And seeing this use of organoids and these new ex vivo models has been really exciting. So it's a very high growth area. And so I've really focused my company on trying to develop the next generation of tools to really facilitate that growth and facilitate scientists to discover these next therapeutics and really do that faster. And so be able to get these treatments into the clinic, into patients, and be a big impact for patients on a much faster basis.
Fantastic. Last but not least, Dr. Matthew, who is really doing some outstanding work down in Tulane, and not only, I think, understands, the core science at a deep level, but really understands the big picture about where cancer treatment is going. You might want to turn down your volume if you're getting some feedback.
Will do. This good? Martin, can y'all hear me? Thanks so much for the opportunity. This is a great forum. I really enjoyed, talking to you recently, Martin, and getting an idea of what and where this is going. And so thank you again for the opportunity to present and talk about what we're doing research-wise and how we've connected with Evan. And as I said last night, this is, I think, is a great way to help share how these results and how these, how our work in the lab and how our work with patients can work with, Mahesh, Evan, and move things forward to a personalized medicine. Maybe I could show some slides. Let's see.
Absolutely. I think you have control over the slide deck.
Okay. There we go. So again, Matthew Burr, I'm an associate professor at Tulane University. I've been a cancer researcher for almost 30 years. And my focus has been on breast cancer predominantly, but we've looked at a number of different tumor types. And I've watched in my career as we've gone from very overlooked issues with model systems that led in many ways, scientists down different and incorrect paths, and how that's progressed to what we're looking at now and it's sort of this personalized medicine space, individualized medicine and precision medicine in combination with patient samples, in combination with model systems like protein fluidics and like a number of other systems that are available moving towards microphysiological systems. Basically, let's see if I can move the slide. As I said, my focus has been on breast cancer, and this is really just a case study. This is a broadly addressable issue across a number of different cancer types. I happen to focus on breast cancer, and when we think about breast cancer, we really think about subtypes. Our focus is on triple negative, but luminal A, luminal B, and HER2 rich. We think about breast cancer as divided into three. Does it have the estrogen receptor? Does the tumor respond to the patient's endocrine system? And if it does, we have therapeutics, we have targeted agents for that, and we have sequential targeted agents and combination targeted agents. Then you've got the HER2 enriched. We know what drives those tumor. It's that growth factor, it's that HER2, and we have a number of agents that allow us to address that clinically with HER2, and that's been developed over the past 20, 30 years. However, this last subset that doesn't have the endocrine receptors, doesn't have HER2, we define as triple negative. And that's really just an arbitrary definition that it doesn't have a target yet until recently. So what's happened with triple negative, it's been sort of relegated to this, we don't have a targeted therapy, so the patients seek chemotherapy, and this tumor type happens to be particularly aggressive, recurs frequently, recurs resistant, and often spreads. And unfortunately, it also targets a younger population. So our research is focused on triple negative, trying to find what are those targets. And what we realized is that as we were moving forward in doing our research, our model systems were limiting us. We had cell lines, but we didn't have model systems that recapitulated what was the tumor doing in the patient. So when we think about triple negative now, what the oncologists do, patient comes in, they have a tumor, it's diagnosed, and they give them what we call neoadjuvant therapy. They're given chemotherapy up front, and at the end, they go to definitive surgery, they remove what remains, and they hope that the patient's had a good response. The problem is, what if the patient hasn't had a good response? Or what happens when the tumor comes back in a metastatic site? You've already treated the tumor up front, you already know it's resistant to one agent, but you haven't gone to the next step and figured out, well, what is it sensitive to? And so that's where the concepts of personalized medicine and model systems that Evan and Mahesh are talking about converge. And that's where our next leap forward, I think that's where our opportunity is going to be. I'm showcasing triple negative, but please understand, anything, pancreatic, colorectal, they all can be incorporated into systems that will allow us to ask these questions. So imagine you've got a patient coming. My wife happens to be a breast oncologist. Her population is predominantly African-American here in New Orleans, and she sees a lot more triple negative than other places see, the rest of the country. So that patient comes in, and all they can offer right now is standard of care. We'll give you a taxane, we'll give you adriamycin, we'll treat you, the surgeon will remove the tumor, and then hopefully it's not going to come back. But what if we could, at the same time that surgeon's taking that first biopsy, that after the oncologist treats with chemotherapy, that remaining tissue can be taken and used for precision medicine. Sure, we know about the genomics. We know that they can be sequenced. We know we can look for mutations that drive that tumor type, or that may lead to say, hey, this should be a PI3K inhibitor or this. But those agents aren't approved in triple negative yet. They're still under clinical trials. But as they move forward, as these agents get approved, not only in breast cancer, but in other tumor types, we start to build a library of approved agents. And then we can, from an informatics standpoint, sequence the tumor, but from a biological standpoint, actually ask a question. If we can take that patient's tumor and say, now we have a panel of 100 drugs, what works? And so if that tumor does come back, or that tumor didn't respond completely to chemotherapy, when that patient goes to surgery or when that chemo is finished, we now have something else in our armamentarium that can be tested or that can go through phase clinical trials and ultimately be utilized to help that patient. So the goal is sort of that dual track approach. We know what everybody hears about the proteogenomics and the transcriptomics, but the biology is also critical, because the transcriptomics is only as good as the site you biopsy, whether that's a primary lesion. But what about that recurrence in the scenario I'm describing? The patient's tumor doesn't respond to chemotherapy. Now we have to go back and look at that again. But there's only so much that transcriptomics can tell us, because that also depends on where you get that biopsy from. And we know tumors, particularly in the case of triple negative, this is a disease of health disparity. Patients don't have, in some cases, access to good health care, to screening mammography. And if these patients aren't being seen early enough, these tumors are getting larger. That means they're getting more heterogeneous. There's more clonal evolution within the tumor. So you may biopsy and get a good transcriptomic profile, but what may be happening is the biology, not only the cancer cells, the immune cells, the fibroblasts, the endothelial cells are all interacting in that tumor. And if you take a cell line out of it and you treat it, you're not getting the rest of the system. So that's the point of convergence where we were. My wife was seeing these patients that were coming in with large tumors, that there was limitations she could offer. So we started focusing our efforts on how can we address that. And so the way we did was we looked at our model systems. Well, we knew here at the bottom.
Let me just interject for one thing, because what you're saying is just absolutely critically important. I think everything is about problem solution. What is the problem? The problem is that we're flying blind as physicians when it comes to treating tumors, because tumors have been thought of as some homogeneity kind of piece of tissue. And that's not really the case. So what we have found over the years is that our outcomes are less than optimal. Even though the outcomes are improving over the years, there is a very significant number of subsets within cancer that become resistant, that don't respond, that metastasize. And then we try as physicians and healthcare providers to come up with our own secret sauce. And our own secret sauce, for lack of a better word, is trial and error. And this trial and error is generally termed off-label use of drugs. And the off-label use of drugs has a very mixed history to it. What I would say is that what we're looking at right now is an incredible set of technologies that allow us to take human tissue, pull that tissue out, and create, in all the senses, a real piece of organ, whatever that organ is. If it's a breast or pancreas or lung, we can create a replica of that. And we have the physiology, the anatomy, the biology, the biochemistry that represents that individual patient. So I want to focus in on this notion of what is personalized precision medicine. Precision medicine is your own cells, your own DNA. And we have now, through companies that we're talking about today and research that we're talking about today, taken the next step forward in bringing all that tissue into a working organoid or canceroid, so that we can test not just one or two, but multiple drugs and combinations of drugs on that tissue to look at the efficacy of that treatment before we do off-label use testing on patients. This allows us to be much more confident about the side effect profile, the efficacy of our treatment, and it may even change the initial algorithms that we're looking at. Now, we're going to look at two aspects of this. The first is what we are seeing now. We're in the first chapter of a 100-page book. And what we're seeing right now is the ability to advance technology, and you will hear from Evan and from Matthew about that technology advancement. In the second part of the conversation, we're going to look into the future, because all of this means that you are going to have more cancer treatment options as patients, that your health is going to be improved, that the side effect profile is going to be improved. This speaks to what we see in Endeavor, and I think what Newchip sees as an opportunity for leaders in entrepreneurial ventures to really take charge and to drive as quickly and as aggressively forward with collaboration partners like Tulane, where science, research, physicians, entrepreneurs, business people can collaborate with large pharma and large biotech. So I want to then shift back over and go back to Matthew. Matthew, you still have a bit of a feedback problem on your side. You might want to reduce your volume a little bit.
All right, I reduced it more. Sorry about that. Yeah, Martin, that's a great interjection. I really appreciate that, because essentially you're correct. What the physicians do now is, when they run out of options, they either go to something off-label or they go to another agent, and it's really flying blind, because you're treating a patient for 1, 2, 3, 4 months until you have the opportunity to check, is the drug even working? And what you're saying here is exactly what the goal is. Can we do that ex vivo and have an answer before, so we know what drug to put the patient on? So talking about the models, we start with this concept of what we were doing 20 years ago was just cells in a dish. And what we realized is, drugs are working great in these primary cells when we go into a mouse xenograft model, but the approval rates for oncology drugs was still, less than 10%, less than 7%, 4.7%. So it's a dismal failure at the back end, and the front end is looking clearly at the wrong systems. And so what we were missing was the increased complexity of a system. And as Martin just pointed out eloquently, these patient samples have everything. You have the capacity to look at the immune impact, the stromal impact, the fibroblasts, all the cells of that individual tumor, and ask that question, what is it responding to? So our goal was, how do we get to more complex systems? So we first started taking those patient samples and going into mice to generate what we call patient-derived xenografts. The advantage is you have an individual tumor in there. The disadvantage is it's expensive, it's time-consuming, it can take months to establish these tumors, and that's not helping the patient. They're great model systems to test agents in, but getting to the precision oncology is not there. So the step we needed was to skip the mouse and develop something where that primary tumor could then be put ex vivo as an organoid, and then established, propagated, and tested so that can benefit the patient in a straightforward and short-term turnaround standpoint. And that's where the microphysiological systems in, and that's actually the point where we connected with Evan, and we saw this great opportunity where we were working with these patient samples, but we needed systems where we could keep them under the right conditions and be able to, without disturbing the system or creating any additional perturbations, ask questions. Does this tumor respond to this drug? And if so, is this the direction we can move this technology so that every patient that comes in ultimately, and maybe I'm jumping to the future too quick, but gets that test? And again, for us, it's actually very personal, from our city. And we talk about access to healthcare. This becomes the next question. As these technologies advance, how can we make these accessible, and how can we incorporate diverse populations, diverse backgrounds, diverse socioeconomic factors, which all are coming into play to provide this technology when it reaches maturation to those populations? And I think part of it is that the approach we've taken is to get those populations invested in the research we're doing so they can see this. And that's why, again, going back, I think this is a great platform.
Let me just say one thing here first of all, just to highlight what you just said, there are disparities within the healthcare system. There are disparities in terms of access, and even within the whole kind of broad sector of diversity, equity, and inclusion, there are lots of subsectors of hurdles that people are up against to get themselves involved in advancing scientific research, particularly through clinical trials. And one of the things that that this allows now is an ability to reach out to the community, to talk to the community, to build trust in the community, to let them know that things that are happening at Tulane and with precision fluidics, those are things that are going to benefit them, and that they should feel that the healthcare system within the community is supportive of what their needs are, and most importantly, they're listening to their specific cultural, racial, gender differences, and that they're recognizing that it's important to be able to bring in diverse groups to test these new models, because the FDA clearly has indicated that the data that's coming out of clinical trials is not sufficient. It does not accurately represent the way drugs and their profile for safety and efficacy reflect real life data. And that's because many of the clinical trials are underrepresented by important diverse populations. So with that, I want to kind of extend out a thank you to everybody who is currently in the audience and watching this, which we do on every show. We want to thank people like Martin, who's actually dialed in from Norway. We want to talk and say a big thank you to Jenny, who's always been involved in a meaningful way, and Chad, who's coming in from Dallas, and Malik, who is involved in genetics from New York City. We want to thank Raj, who's also a big participant in many of these webinars. And we want to encourage people to please log in. Tell us where you're logging in from, and please give us comments about what we're talking about, which is the ability to improve healthcare for everyone. Most importantly, to change the way that healthcare providers look at solving problems to improve outcomes. So with that, I just want to throw it back to Matthew. I know you have another slide up here, which I think is a reflection of some of the things I just talked about.
Yeah, summary of the approach we take from patient to sample. And again, this is sort of a research diagram, and this is the kind of diagram that is great to show what we can do with patient samples, but this doesn't represent what this does for the patient fully. And that's sort of where we're going, and to ultimately get to that patient first approach, that whereas the research that we're doing drives the research, finds the targets, for the next generation of agents, but at the same time, in tandem, to have that benefit the patient directly. And that's sort of our goal. And with that, I just wanted to show you an example, which is just how we create these canceroids, these tumoroids. In this case, this is a patient that had a triple negative breast cancer that received neoadjuvant therapy and did not respond at all. The tumor continued to grow. The surgeon was able to remove the tumor, but unfortunately the disease was very advanced and very aggressive, and this patient succumbed. We were able to establish tumoroids and explants and PDXs from this. And what we're showing here is that the spheroid, it does exhibit sensitivity at the highest levels to taxane, but at the lower levels, which is what the patient, in the range the patient was receiving and did not respond to. So we've then taken and later published this data and showed that we could take these spheroids and then go back to those FDA-approved oncology drugs and ask questions about, well, are there agents that may have impacted this tumor? And so that's sort of the goal, but to do that in real time with samples. So with that, I'll turn it over to, and like I said, this is data that we've generated with Evan and in collaboration, and his reaching out to each other really opened up a lot of new approaches and avenues. Because I guess what's great about this is you tend to sort of get stuck on your own thought process. I know there's something else. I know there's something I need, and I haven't found it. And then, we came across Evan, we started talking, and it blossomed into the direction we're heading. So thank you.
Absolutely terrific beginning of the conversation. I don't know, Mahesh, before we go to Evan, you see so many companies, you look at things from a big picture as well as, from an operational standpoint. The impact that I see, as part of your team, obviously is siloed in my world, which is I'm a physician. I'm all about, patient-centric care. From a company perspective, can you tell us a little bit about what you see in terms of the personal stories or the personal enthusiasm that you see driving more healthcare entrepreneurs and a greater flow of these companies into Newchip?
Yes, absolutely. I mean, I think having been on both sides, where we're helping companies, but also investing in companies and through both of our funds. I think it's important from an investor perspective that the founder is passionate, that he or she understands why they're doing something. And oftentimes, when it comes to healthcare, and this is very common amongst many of the founders that come through Newchip, but I think it's in general, many of the reasons for why we get involved in this space is because not only is it the educational background, but maybe we know someone who has been diagnosed with breast cancer. We know someone who's been diagnosed with prostate cancer, lung cancer, colorectal cancer. All of them, in various levels, have some type of need, and that's one of the things I think that drives a lot of entrepreneurs, and often who we see succeed as an entrepreneur in Newchip are those that are driven not just by knowledge, not just by a great product, by having the passion to go solve that problem. And so I think that's a very important component to not just healthcare, but to any problem. But I think more often than not, I see that as a very common thread amongst many founders in healthcare. And that being said, when it comes to the opportunities that are available, and I'm going to leave to Evan to kind of go through the advances we've had so far. But there's so many opportunities in synthetic biology, in organoids, in drug development, especially as we move more and more towards precision care and personalized medicine. Being able to identify those differences between two seemingly identical patients, but those genetic differences are what changes the makeup of each of their genetic markers, their phenotypic markers, why certain people respond to certain drugs, not others. And there's still a significant opportunity there to not only come up with solutions, but also identify how the solutions can be developed for each of these individuals, and make them better, make them better responding. I think a big part of maybe the past 5 to 10 years has really been turning these quote unquote cold tumors hot. How do we get people who are not responding to certain treatments actually respond? And I think a lot of the research that I've done in the past, including my graduate work, has been in immuno-oncology, having developed certain treatments like CAR Ts and monoclonal antibodies. I think these are very, as you mentioned, these are very early steps. These are the first chapter of that much longer book that we need to write, where we actually can address each of these issues personally, be able to address it in a more precise way, so that we're getting better outcomes from patients. And I think what Evan's building right now takes us that one step further, where not only can we identify if certain drugs are working on certain organoids based on their own tumors of those patients, but we can also identify what are the side effects. We can see where is it working, but also where should it not be working, right? And a big part of, I think, precision medicine is that we have had issues with off-target toxicity, but now we can try, negate that, or at least mitigate that in some way, using these organoids and seeing how else are we affecting other parts of the body, other patient populations. As you talk about diversity, it might work for one population and not others. And to Dr. Burrow's point, something like triple negative breast cancer highly affects certain populations than others, and then yet we're testing it on one population and not really the population that's really being affected in a major way. So I think those type of understanding of the patient population only through organoids, I think we are able to create better accessibility of these drugs, but also getting better results of what where will this work? Where will this not work? And I think this is a great step forward, and I think there's a lot of opportunities to make this even better. But I think Evan and his team, as well as Dr. Burrow's team, they're in a great track to get us there. So I don't want to take too much more time. I do want to hand this over to Evan so he can actually explain the advances they've made and really dig deep into where this is going to grow in the next 1 to 2 decades, and where I see tremendous opportunities for actual treatments, actual cures for cancers and many other indications we just haven't had a way to get into than how we do.
Yeah, no,
definitely want to. Oh,
go ahead.
I was just going to say, I want to come back to what Mahesh said about immuno-oncology at the end, because you made some great points there that I think are worth discussing in the context of these systems. Thanks. I'll let Evan.
Well, one thing I just want to add here, because, that was a tremendous overview. We have different people from different perspectives contributing, and that's the point of having this panel. Here, just an incredible depth of knowledge from the entrepreneurial side, the leadership side, the scientific side, the vision that's embedded in this is fantastic. I would overlay one other aspect. And, we at Endeavor formed a venture studio in June of 2022 with the focus of developing leaders who want to collaborate. And, part of this process that we're talking about is changing mindsets. Changing mindsets means trying new things. And when you try new things, there's always a need for collaboration, partnerships, team building, communication. That's really embedded in a lot of what we do in the venture studio. So we, like Newchip, we really look for leaders. We look for entrepreneurs who want to participate in a team effort, who want to hear different people's perspectives and ideas and say, you know what? We can do this better if we bring a few voices together. And part of what we do at Endeavor is we bring in early biopharma, med tech, health tech, biotech, even in the seed stage level, just to observe, just to learn what we're doing in a transparent way. We host workshops with Capgemini and other large important tech players like Microsoft and Amazon, just to show that we can bring together like-minded people who then come to the table. As we're seeing here, there's a collaboration that spawned a whole new generation of research between, Evan and Matthew. And so that is exciting to me, because to solve these problems requires partnerships and collaboration between small companies, large institutions like Tulane, and large corporations. And so, I am very bullish on the next decade in terms of science, because I do see within certain models of investment, like venture studio models, the ability to unlock a lot of value for new leaders. With that, one of the new leaders or continued leader is Evan. And I would love to turn it over to Evan, because, what you have done is, you've broken ground and you've moved the whole field of cancer research forward with Matthew. Tell us about what your company's mission is, and tell us where you are now, and then we're going to transition into where you see the company going.
Cool. See, can I advance slides, or do I have to present myself?
You can advance slides if you give me a moment. Try that now.
Okay. Excellent. Yeah, so our company mission, as I talked to you, my passion is really tools, and my background came from physical chemistry, where I was developing really complex tools to analyze materials and do materials analysis. And I shifted into biology about 20 years ago, because I was really fascinated by the cells and biological materials and live things, and all the things that you could take these tools from other industries and really apply them to a critical problem, and helping scientists like Matt and you, Martin, all the others, physicians out there and researchers out there who want to understand the biology better, but need better tools. They need better ways to look at their samples, need better models, and What we do is bring those models into the lab. The first system that we brought to market, which is commercialized in Matt's lab, is this Puma system, what he used to do the work on this triple negative breast cancer. But where we really think this is going is trying to help scientists and physicians, understand cancer better. I think Matt, you talked about this, how, currently, if you have a cancer patient, you look at the genomics markers, you look at pathology, et cetera, and you'll try to determine the best course of treatment. But what if we could give you better information? What if we could come through and not just look at the, tip of the iceberg, but give you more accurate drug choices, patient-specific information, give you fast results. And so instead of waiting 2 or 3 months for results, wait 2 or 3 days, and then you can use that an array of samples, et cetera, to really pinpoint what's the best treatment for this patient and what's things you want to avoid. How can you successfully cure your patient?
Yeah.
So the future then we feel is we call our predictive system, or Puma Predict, where you can take a patient sample, take the biopsy, turn that into an array of samples, where you can now do multiple different treatments. You can use patient-specific results, and you can get better accurate data. And one of the things we're doing by moving into the future is doing 3D imaging, 3D rendering of these patient samples and looking at phenotypic responses, functional responses of the patient's samples to treatments. And by looking at those functional responses, you can then use advanced image analysis. You can advance machine learning, artificial intelligence to this complex data set and give faster responses and faster answers to the patients. So again, this is all about how speeding up this process, making it patient specific so you can do precision work, and making it accurate, predictive, so that the doctors are confident when they take the samples, and researchers, when they're trying to develop next generation therapeutics, can do that in a much more efficient fashion.
So first of all, that's just an incredibly, deeper level of understanding. I think the slides are very descriptive about the process, and your personal involvement as an entrepreneur, as a scientist who's, I'm sure faced many headwinds and moved this forward, to advance cancer scientific research is just outstanding. What I would say, as an investor, looking forward, what I see is that companies now are looking to engage with emerging companies like yours. What does that mean specifically? So one of the concerns that almost every company has is financing. There is this notion from CEOs, Mahesh is shaking his head, he understands. Every single CEO thinks their company is going to fail because they've got 6 months left of cash and they're in crisis mode. And that crisis mode never goes away. It's just a vicious flywheel. The problem is, and I wrote an article about this in Fast Company in late January, that it's a horse and cart situation. You need to build a great team. You need to build a great company that has vision and that has collaboration partnerships before you make the case that your company is valuable. And in this sense, I'm kind of pounding the table for the notion that, working in the environment that you and Matthew are working in Tulane is a call out for collaboration partners. It's a call out to large pharma and biotech companies to come and work with you guys. It's a call out to other entrepreneurs to roll up their sleeves and say, wow, what you're doing really fits in well with what I'm doing. So I would encourage people who are listening now and people who are going to be looking at the recording to really see, is there a fit between what you're doing as a company and what Matthew and Evan are doing? And the reason is that in order to deal with complex problems, you need to bring in partners. You need to bring in collaboration partners who have strategic ideas, and then you need funding. And I think it's a question right now in a very tight economic environment about how do you not let great research, great companies die on the vine. I'm committed, and the venture studio is committed to funding companies from seed all the way through Series B. That is our commitment. We take 4-year commitments very seriously with the companies that we work with. And now with new collaboration partners in large pharma like Sanofi and others who are creating incredible accelerator programs, who are writing checks of 230, 370, $540 million for emerging companies. There is now a pipeline that should empower CEOs to understand that money is fungible. People are not. Bringing great people together to build great teams brings the money. It's not the other way around. It's not that you have a great piece of innovation and that people will come running to you. It's that you have a great piece of innovation that's going to affect human beings, and that you have a team that can execute on that, and that you have collaboration partners who consistently work with you to be resilient, improvise, pivot when necessary, and accelerate when other people are fearful. I see Mahesh shaking his head, so I'm going to throw it over to him because he's on the investor side.
No, absolutely. And I think, Martin, you put it in such a great way. It's important when investors are looking at this, especially in the early stages. Many of us are looking at the team, even more than the product. The team is really what we're focusing on, because we are believing in the team. We're hoping that the team here that we're investing in is going to move this forward successfully, able to take it to growth stage, able to get it past the regulatory approvals if needed, and then hopefully commercialize and do well with the partners that you mentioned, big pharma, small and large biotechs that are able to use these tools to then, better their own research, better their own results with that research, and come up with great innovative drugs, innovative diagnostics and tools that are going to benefit everyone. And so having that team is very core, especially in these early stages of investments. But as they grow, having those partnerships, having those collaborations with larger pharma, saying, we have a very clear path to commercialization, a very clear path to potential, acquisition or, merging with a larger player. These are all providing comfort for investors as well, that there is a potential exit opportunity. Because as much as investors are investing in these great products to better our lives as a species, they're also looking for returns. Right? We are also looking for returns, and having these collaborations also enable that conversation to be a lot more straightforward. Saying, okay, we can see that as you mentioned, Sanofi or one of the other larger players, having a collaboration there, saying, okay, they could acquire you, they could potentially license this product from you. And so we see a long-term play with that collaboration, not just as something that they can do now, but having the potential to have a exit, have a in a long-term perspective on returns is very important, because even more than other industries, deep tech industries like healthcare have high returns, but at the same time high failures as well, because oftentimes it's binary. It either works or it doesn't work. There's often never a gray area there. Either you get FDA approval or you don't get FDA approval. So when you have such large margins within you being right and you being wrong, it's very important for the team to be core, to have those collaborators who can help you move past those pitfalls, make sure you're successful both on the regulatory side, on the commercialization side, and then on the investment side as well. All that's very important. So again, I kind of expanded on what you said, Martin. Hopefully, that kind of comes across as more of a need for teams that when we're looking at investments in the space. It's not just the technology, not just the indication, but the team's going to be very important, and who they are teaming up with as well. The collaborators are also going to be as important.
Fantastic overview. I want to add a couple of points and then throw it back to what the vision looks like for Matthew and Evan. So I want to highlight just a few companies which go towards the collaboration focus. These companies, Flatiron Health and Komodo, both look at outcomes. They look at analyzing big data. They take that big data and they see, what does this mean in terms of the impact on patients? That's the future of healthcare. those are the kind of collaboration partners. As we collect more and more and more and more data, they will play increasingly important roles. And then the second, is a company that I personally involved with and support wholeheartedly, which is called Clinical Trial Hero. They focus in on improving the speed in which underserved patients who are minorities, patients who have a gap in terms of their ability to enroll in clinical trials and enroll in cutting edge medicine. They register these patients with a focus on addressing their cultural needs, addressing their specific gender needs, addressing many things that often medicine as an institution ignores. Bringing together organizations like Clinical Trial Hero, Komodo, Flatiron Health, in collaboration with Tulane, in collaboration with what Matthew and whatever Evan are doing, are things that I personally would love to be able to bring to you if that's something that the team wants, because this is an incredible story that has really yet to blossom in a national way. And so that offer is on the table. Let me throw it back to Evan and to Matthew. Let's spend just a couple minutes because we're almost at the top of the hour. This has been such a dynamic conversation. What do you see as the next 3+ years out? Like, where are things going in this canceroid organoid development so that we can create an ability to test more and more combination drugs, different types of drugs, in two specific ways? First, it's my feeling that by addressing the environment of the tumor, we're recognizing that we have some success in a very broad way with broad chemotherapy. But then there are microenvironments within the tumor itself. And we cannot be heavy-handed about addressing these microenvironments because the side effects of the first-tier drugs are so severe. So my personal vision and my question to you is, do you see a role for being able to be smarter and more effective with fewer side effects in addressing, let's say, the systemic problem, and then being a little bit more focused and heavy-handed on that microenvironment where we know that specific cell types become resistant? We know that different cell types have problems in terms of essentially reacting positively to certain types of chemotherapy, so that we can mix and match multiple drugs to address those microenvironments in a stronger, heavy-handed way. I'd like to open that up to Matthew and to Kevin in the few minutes that we have left.
Yeah, no, that's great. I mean, there's a lot to say there, and I'll try to go quickly. I think that the future over the next 3 years is going to be adapting these technologies towards immuno-oncology. I think Mahesh really captured that when you asked the question, how do you make a cold tumor hot? I see as a double track here. One is, so for example, in the case of triple negative, Pembro, which is the anti-PD-L1, was approved recently or several months ago. It's a very tough regimen for a patient to go through in triple negative, which, you've got your platinum, your anthracyclines, you've got your Pembro, and it takes a long time. When it works great. When it doesn't, the side effects can be horrible. So something like a platform where you could ask the immuno-oncology question, something you can't do in a dish and something you can't do effectively in a mouse, then we know, okay, is this patient going to be predicted to respond or not? Then you move forward and you ask the questions like you're saying, can we combine it? So the research arm to that question is, let's say I have a patient sample that doesn't respond to immune therapy. Can we start to look at combinations? Can we start to get away from this heavy cytotoxic regimen where you're adding a targeted agent on and get to something where we modulate the immune system, and then we really go after those in those tumor microenvironments that respond? The TME is critical. That's what we have to target next. How is the microenvironment protecting, impacting, feeding the tumor in a way that leads to resistance, or a way that leads to failure for the immuno-oncology to work? I mean, this is exciting stuff, and yeah.
Evan, you have the last
question, but
no, it's a fantastic and I think impassioned description. And I'm all in for coming down and exploring some jazz and perhaps having a bourbon together to expand upon what we can do together. Evan, you have the last few minutes here to talk about what you see the role that your company has in the next 3 years as this moves forward.
I see, one of the biggest impacts of the technology we bring to the table is really in the immuno-oncology area. Because if you think about, chemotherapy, you have a solid tumor model. Yes, you want the right tumor microenvironment, but at the end of the day, the chemotherapy is a small molecule drug that's interacting with the cells, and you're looking for cell death. Immuno-oncology is a very dynamic process. Now you have immune cells coming in, interacting with this model. Cancer has developed resistance to immunotherapy over hundreds of millions of years, I guess. But from an analytical standpoint, tool standpoint, watching those cells come in, looking at how they're interacting with the tumor, how they're being resisted against, how they're being pushed away, or how they're attacking, and understanding that dynamic on a very intimate basis is, I think, going to be a huge game changer for immuno-oncology, and being able to do that again with patient specific samples in a dish, so you can look for on target, off target, what works, what doesn't work. But that whole dynamic interaction of immune cells and 3D tumor models is a very amazing system, and I'm really excited about bringing tools that can help researchers understand that in a much better fashion.
I want to thank everybody for an incredibly extended webinar today. We could talk about this all afternoon. First, I want to thank all of the participants here for their incredibly insightful comments. And second, again, I want to circle back to the people that attended from all parts of the world. Thank you so much for being a part of the Better Health webinar series. Please tune in over the next coming weeks. We have some exciting companies, including some of the companies that I just mentioned, like Komodo and Flatiron. We're really looking forward to your feedback and your comments. Please let us know how your personal experiences are being changed by some of these webinars. We do have a subscription to our newsletter just by emailing us at subscribe@gershoncapital.com. You can also include comments or thoughts or suggestions about companies that you would like to see on the webinars. And finally, a shout out, to collaboration partners, emerging companies, large companies. We would love to be talking with you and have you as featured speakers, because this is about building a community and about affecting change and outcomes. So thank you everyone on the panel, and thank you everyone in attendance. Have a wonderful rest of the afternoon. Panelists, if you can hang on for a couple minutes after I end the broadcast.
Thank you, Martin.
Thank you so much.